migration, physiology, and behavior with her, my mentor and biology hero. So with no further ado, Marilyn is going to talk to us about migration studies through the lenses of migrant and resident comparisons and seasonal stages of spring and fall.
Thank you. Well, good afternoon, everyone, and get started. So, first I want to thank Jamie and Suzanne and Bob for being, and Tresha for being such great hosts, and for inviting both John and I here today. It's been wonderful to be on campus and meeting all these wonderful students with wonderful projects. It's a great place. So I'd like to get started, and as Jamie said, we're really looking at these two subspecies of wood-crowned sparrow, and I'm very interested in knowing, are these subspecies same or different, and throughout their life histories, throughout their migration life histories and resident life histories. Let me get started. I'm missing the title. I'm just going to go back, please. Up here on your own. This is what she. What is this? Okay, here we go. So I'd like to go to my introduction. Migration has had a deep history of scientific investigation into behavioral ecology, physiology, navigation, body level, lots of things. And most recently, studies on movement have been just stunning. That is, patterns and distributions of migratory birds. And this is largely due to technological advances. It's just amazing. On the other hand, the basis for the physiological and behavioral mechanisms has not kept pace. And there are people working in the field. Jamie is one. John has done a lot of work, but we need to pick up. And so our work has really focused on these steps. And so what I'd like to do, two things, is look at a two-pronged approach. Talking first about the environmental influences of the migratory life history, taking a life cycle approach to compare the seasonal traits of the spring and fall migration. So this is within a migrant itself. The question is then, are these traits the same and different, and what does that tell us about the selective pressures on migrants during the spring and fall? For a long time, people thought, oh, they're just the same, they're just going in different directions. But now our ideas about that are changing. And more historical perspective I'd like to discuss is the migratory strategy, and compare the traits of a migrant and a resident to understand what features have been retained or lost over time. And the question is, are these traits the same or different? And what does that tell us about each of the migratory strategies? To start with, let me just define what migration is, because migration means a lot to a lot of different people. Migration is the movement between distinct habitats, one generally for breeding and the other for non-breeding or overwintering, and each offering seasonal benefits or resources that increase the fitness of an individual. So that's movement, purpose. The other things, is that showing? Oh. So what I'd like to use is the ideas of the concepts of life cycles. John presented this yesterday, and it is an area that we can really use employed in migration work, and it's helped a lot. And it was the point that Jamie made, that we're trying to understand each of these small segments, these successive stages that migrants go through. But not Okay, so these are concepts of life cycles or phenotypic flexibility. So nearly all organisms live in fluctuating environments. We know that, we live in that every day. Organisms can really alter their states to maximize the survival at any time of the year. So they're changing, or they're showing phenotypic flexibility throughout a stage across the year. Several. And then we've taken these ideas further, and we're saying that these alterations can be summarized in a temporal sequence of life history stages with an overall benefit that maximizes lifetime fitness. And this has really helped us to create a concept of migration that we can then go in and test. I'm sorry.
It's not you, Marilyn.
It's not you.
That's what it is. Can you advance on you?
It's okay, going on, I guess.
Yeah, it's something here. It's not in presentation. Bottom right.
Yeah.
Bottom right. Yeah.
Here
Yeah.
you go. That
Oh, yeah, there.
Hey.
There it is.
Yeah, I
Okay.
think that's confusing it, so.
Okay. Okay, so these are the players. This is Zonotrichia leucophorus, and on the left here is the migrant, that's Gambelii, long distance migrant, and its subspecies friend is a resident that is Zonotrichia leucophorus. And we thought that these subspecies, and probably other ones in this whole plane, have separated from the common ancestor over 50,000 years. So they've been separated for a long time. But they did arise from a common migrant ancestor. Today, the ranges are such that the migrant reaches far north of 69 degrees. And John was presenting that was dated yesterday. They winter throughout the West from the lower 48 all the way to Mexico. So they have a huge range where they overwinter. And they show about a year-round mileage of 8,000 kilometers. So then they're on the move. And when you've got that much movement in your life cycle, there's not a lot of time to mess around. So they're really constrained in terms of time and on the other hand, the Nuttles, which is resident along the northern California coast. And doesn't move in these areas or on this one location for lifetime. So they have a very different life history. And I just show this cartoon to give you an idea of what these movements are like. So in the wintering stage, our study sites are in Yolo County. However, for Gambelii, they are spread out up into Washington and then all the way down to Mexico. So they have a huge range, but here Nuttles are right along the coast and they remain there. In spring, the Gambles showed an outward migration, the spring migration, into areas in the Arctic, into Alaska. And this is work we did with Simeon Wazowski, where we had geolocators. So we actually know for a few of them, not the whole population, but they're breeding in these areas in the southwestern portion of the state, all the way up to and beyond the Brooks Range. And these are the stopover sites. So these are overland short mountain migrants. And so they will fly by night and stop in areas to refuel during these periods of time. On the return, we were pretty dumbfounded, but it's not really that surprising for people who are reading in terms of migration literature, is that these birds, all of them arising from their different breeding sites, head east into the Yukon on their route south during fall migration. And they tend to stop for longer periods of time, many more stopover sites. And then they return to their wintering grounds. They move down the east side of the Rockies and then flip over, coming down the Cascades into the Sierra, and then down into the breeding areas. So we know that there are differences in terms of routing and in terms of timing during these two periods of winter. So what I'm going to do now is introduce sort of the life history stages of the two. On the top is the migrant life history, and you can see it's busy, and it's got a lot of different stages. And so we've identified this as having high finite state diversity. So there are a lot of stages spread over the 12 months compared to the resident that has essentially 3 stages: wintering, breeding, and molt. So you can see right away that the times they spend throughout the year are very different in terms of the number of stages and the duration of these stages. The question is that how do you get a migrant from its wintering stage into these very complex stages of vernal spring migration, breeding, etc. And one thing about the zoonotrichia, or most of the zoonotrichia, is that they're highly photoperiodic. That is, they rely on photoperiod, particularly on the spring side of things, to induce changes. And so as here's an increase in day length, as day length increases beyond the spring equinox, this activates the hypothalamic pituitary gonad axis to induce hyperphagia, fattening, and changes in mass and optional behavior. So this is then the induction of vernal migratory traits that also induce breeding. So you've got these couple of events that are induced by photoperiod. And also, this photoperiod also affects the pre-basic molt, but actually onset of the autumn phase, that would be autumn migration and return in the fall, are not under photoperiodic control. In fact, the birds are fractal. So we know a lot about the spring. We know quite a bit about the spring phase, but very little about the autumn phase. So to say they're the same or the different, we really don't have the data in hand. And the point here is that with the resident, we can see that breeding is actually starting as early as January, at a time when photoperiod is still very short. So we think they're photoperiodically driven, but it's a very different regime. It's a very different system. And Barbara Blanchard years ago was saying that these nuttals are starting to breed as early as January. And for people working with animals, they said, well, that's heretical, that can't be. But it can. So what I want to do now is step back and look at changes generally over the months, and then I'm going to flip into using the stages. But I think this array allows you to see what's happening generally throughout the period of spring migration and fall. And what I've done here is that the graph is basically the look at migration. And so we did not do, we weren't sampling during the breeding phase. So that's why there's this cut here. And so we're going from January to May, and then from August until that's what that is about. But here we have displayed the resident, this is the male, female residents, and then the migrants are beneath. And what pops out here is that the residents are bigger birds. Yeah, not surprising. When we look at FAT score, then we begin to see that the migratory period is popping out. This is the male and female migrants, and you can see during the month of April, they're starting to fatten. And the interesting thing is females arrive fairly fat and retain their fat through early breeding, but males drop as soon as they arrive, they become territorial and they're visit-dependent territory. But the point is that the spring phase, in terms of fat and less so body mass, it appears very different than it does. And turn to testosterone levels to give you really a feeling of what the length of the reproductive periods are. So these are levels, and you can see that testosterone levels are actually starting to increase fairly in January, and that period of breeding is quite long. And for mammals, very, very sharp peak, essentially a month, almost 2 months. So those life history stages are very compressed, highly timed, and constrained. I want to just look at it, look just for a short period of time at corticosterone. John went into great detail about it yesterday, so I'm really kind of off the book. But we did look at corticosterone because it's a fascinating hormone. It's complex, it's maddening at times, but we know that among the things it does, it's involved in metabolism, it's involved in behavior, it plays a role during migration, it plays a role during breeding. And so I just want to show you the comparison across the months. And we do this in relation to the stress theories that John presented yesterday. The very quick sample within 3 minutes gives you a baseline measure, which is measured for during the unperturbed period. So it gives you an idea of the measure of energy utilization or energy state of the organism, or the measure of allostasis. And then sampled over a period of time. Generally, we just take it out to 30 minutes because we know so much about the stress series in mammals and white-crowned sparrows. And this induction then gives you sort of a measure of the reactivity or the response to the perturbation. There is no change during the daytime hours, so it's not a diurnal change. It is a response to capture. And then these are the baseline stress-induced levels over the months. And the thing that popped out to us immediately was this very high level in January in female residents. We didn't really expect it. And if we look back in time, in the fall, we can see that it has been creeping up. And talking with John and with Jesse, who have done a lot of this work, saying we realized that the fall and winter months along the Pacific coast are probably the most dramatic. The windiest period is probably the most difficult time for these resident birds. And perhaps the females, as they're beginning to initiate breeding, are feeling the crunch with an increase in allostatic load, an increase in baseline levels. For the migrants, the peaks are occurring during spring migration, much less so in fall. And the response, that is the 30-minute response, we've lost the peak in terms of females, so they're not reacting to capture stress to the degree that the baseline levels might indicate. And so the response levels, migrants are increasing during this period of time. So those are the monthly comparisons, but I'm going to quickly move on to focusing on the it will go. Just so I'm sorry. Okay. So now let's focus on the life history stages of the migrant. So these are just the migrants. And the question is, are these behavioral and physiological traits the same, or are they different? Can I?
Yeah, it should come back. Yeah. So sorry, everybody.
Seems like sometimes just clicking on the slide once
Oh, there we go.
teaches it to.
Click on the slide.
Yeah.
So I got it.
So can you just like this?
Yeah.
Okay.
No, now you can use the.
Now you
Yeah,
can
well,
use it. Now
but it happens again.
Yeah.
So I don't need to do this, but I'm just going to review that. Here we are, looking across the life history stages, and I've done that stuff. So let's take a look then at these life history stages. And what I mean to say is what we found that during this pre-alternate molt, it occurs over about a month, a period of 2 months. The first month, we're seeing early stages of molt. By the second month, that is March, mold is highly intense, so we can actually see a significant difference in the intensity of the mold. So I've actually split them between early mold, late mold. These birds then are preparing to depart. We don't know exactly when they're going to depart. We know it's within this window of the 5th to the 16th of April. So we're calling these the spring departure values. And then at arrival, we catch birds as soon as we see them in Alaska. And these particular days were actually collected in Fairbanks, Alaska. So let's look at body mass, and you can see that it's not terribly dramatic. We're actually seeing a slight increase from molt to spring departure, but not much change in fat. We look at fat score. Of course, fat is one of the primary fuels used during migration. It's a cheap fuel. It's stored without water, so it's light and it's packed with energy. So birds deposit fat in great quantities prior to departure, and that's what we see both spring and in fall. And this is a trans-Saharan market. So these birds, the European warblers, so they are crossing the Sahara or crossing great oceans. And the amount of fat they deposit is really a reflection of how much they're going to need to get to their destination. White-crowned, which are overland migrants, deposit fat, and it's visually apparent, but it's not in the system. What I'd like to move on to is some studies that we've been able to conduct with being at Davis, which is a large muscle group. And we started looking at the pectoralis or the flight muscle. So here's a sort of a long view of the pectoralis, and the pectoralis major and minor. And those are the muscles that are really responsible for this. So the flat and flight are under the auspices of pectoralis muscle. And if you look here, we were interested in trying to understand muscle hypertrophy. And if you're reading in migration, people are measuring various types of hypertrophy. An increase in muscle mass, or an increase in the visual score of muscle, or the increase of thickness, or the increase of an ultrasound measure. So there's lots of different measures. And the problem is that people measuring them are seeing changes at different periods of time. So yeah, we thought that if we could actually just look at the fibers themselves, sort of the source of the hypertrophy, it would help. So working with this muscle group and some very, very capable undergrads and graduate students, we were able to develop a laminin stain that stains the endothelial cells of the fibers. Okay. And what we got is a very uniform picture. So in terms of size, it's fairly uniform. In terms of fiber type, they are all oxidative fast twitch fibers. So you don't have any kind of mixture in here that you often see in other muscles. Let's look at the data. And what surprised us is that across all these stages, except early molt, it's very uniform. So it's not changing. I thought, well, are we getting hypertrophy here? But look at during molt, we saw not a significant, but a decline in these early molt samples, and then a rebound late molt. I can't tell you why. It's interesting. We're thinking that maybe the impact or the energy required for initiating molt is part of this, or maybe this is part of the whole photo induction, or maybe we just don't know. But definitely the hypertrophy is starting for white crowns in late molt, and then it's sustained throughout. When we look at muscle mass, just the whole weight of the muscle, what we see is a slight stepwise increase from molt, and then another increase at spring departure. So and not much change here in fall. So when people talk about muscle hypertrophy, and think of the anabolism that exercise induces. So with training, muscle increases, muscle fiber increases. Also, So that when birds start to increase in mass, a lot of people have shown just the increase in mass in birds in cages, they'll show increased muscle mass. Or increase in muscle, in body mass, you'll see an increase in these. But training itself, flight itself is going to have an impact. You go into the gym and you work out steadily day after day, what happens to your muscles? So we've got effective training, we've got attractive weight, and then there's another aspect that we were really interested following up, and we did so with Evelina Patman, Suzanne. We found that androgen has a huge impact on, at least androgen's cell cycling has an impact or is related to these muscle increases. So androgen, which we know is increasing during these early stages, peripheral androgen is increasing in early stages, that may be contributing to the muscle hypertrophy. What's happening in the fall, we're not really sure. It could be that some of the, just the cellular changes in androgen might be having an effect, or very little at all, because muscle mass is increasing that much. The other aspect of muscle hypertrophy could be due to the increase in changes in myosin heavy chain isoforms. So within the myosin complex, within the contractile unit, myosin has these different isoforms that could affect contractility. And working with Kenneth Walsh and Brandy Belton, they identified the presence of an adult FAST1, which has been identified in chickens, so that's well known. But what she found is that in winter and in decreasing proportions was a FAST2. So this is a novel isoform that hadn't been seen before. And why it changes in proportions, is it contributing to muscle contractility or to muscle flight performance? We don't know, but it is an intriguing change. Yes, that worked. Next, I want to look at some scores that we have used. We developed these in the field, and it's really helpful because you never know exactly when you're tracking migrants, when they come, when they arrive, and when they're going to leave. And what we noticed while we're working with them is that muscle profile is helpful, as well as muscle color. So people have looked at muscle profile as a measure of different states, or can be actually used as a measure of migratory preparedness. And the profile is just a visual measure of the amount of pectoralis muscle that is surrounding the diaphragm here, and this is the keel. So in the zero state, the keel is sticking up above, and you can actually feel it with your thumb. So when you have a bird in the hand, you just feel that keel sticking up, you know, that there's not a lot, and the muscle is very thin. But with hypertrophy, the increase in muscle fiber size and muscle mass, the muscle layer increases and it becomes big, and it's almost like a cap. So it is a fairly clear measured parameter in this. And what we see is that as with muscle mass, the muscle profile increases, and we also get an increase in the fall stage. Muscle color was something that we just picked up serendipitously, and then we noticed that around the time of departure, so early April in Davis, we were looking at the muscle. Normally, the muscle looks like this. It can have some fat at the top. That's a fat store on the top, but it's purple. It's thin. It looks like stick. So it's a darker purple. At departure, around the time of departure, it's pinched up a lot. It's the color of rosé. And so we're giving that a score of 3. And you can see that at the time of departure, our field data showed that yes, the scores dropped to 3, both in spring and in fall. Talking further with muscle specialists at SICK, who I happen just to be riding an escalator with, talking with Richard Dillon at the University of South Carolina, he said, well, if you're interested in muscle, you should come talk to Marilyn, which I did, and it was valuable. And we ended up looking at electron micrographs of these muscle at these various times. And what you see is in winter and pre-nuptial mold, these are the muscle fibers. These are largely mitochondria, which are really interesting. They're changing, but we haven't tracked them that closely. What we were interested in is this spring departure, spring arrival. And these white vacuoles here are actually lipid flows. So you can see at spring departure, these lipid droplets have increased in number. And at the end, they're gone. They've been used. So depositing lipid in muscle puts it really where the buck should be, because then the lipid is altered into fatty acid and shuttled into mitochondria, or fatty acid oxidation takes place. So this is occurring at this time of the year. And it doesn't appear, although with our migrants, it appears that it's associated with fattening in general. But I just read a paper on Aphocephалus sedulars who were looking at muscle fat deposits in relation to muscle, into fat, the body fat, and they found no relationship. So there are times of the year when the muscle is doing this, is depositing fat, that is not related to fattening per se, but might have something to do with the life history stage, or maybe something to do with the occurrence of taking off the unpredictable. I mean, you think when you're taking off for a migratory flight, you're unsure as to what actually is going to happen heading to that stopover site, but having an extra amount of fat in the muscle is certainly advantageous. Then we went on to look at the gastric medius, which is another skeletal muscle and of the leg. And it, if you look, so that when it contracts, it brings the leg up. So it is one of the many muscles involved in locomotion. So not flight, but terrestrial locomotion. And this is the laminin stain cross-section of the gastric medius. The muscle fibers are bigger. The muscle fiber type are much more mixed. They're not all oxidative gastric fiber. And if you look at the fiber area of the gastric medius of the migrant, you can see it's a very different pattern from what we saw in the pectoralis. And this increase here, I think, has to do with the birds are finished with the flying portion of their cycle, and when they become resident, when they are on the wintering grounds, they're not flying a lot. They're flying short distances, but it has nothing compared with what they do. Gastric medius mass, even though we're seeing this tremendous plane, we're not picking up much across the mass. The muscles are really small. So, but if you look at something like a diving duck or a goose, the gastric medius plays a huge role. And there's a lot of changes that we wouldn't see in pasture. So let's review what we got up to this stage in terms of the seasonal comparisons and migration, like this stage. So the outward and return routes, duration and stopover differ, and they're faster in spring. The endocrine measures are more prominent in spring than migrant. Fueling, it seems to be similar. So it does make sense if you're taking off and long distance flight, you have to at least leave with good stores in place. And power flight muscle is greatest, shows its greatest change during the spring. Okay, let's then move on to the subspecies comparison. And this created another sort of very conundrum. Once again, we've got these two arrays. And what we want to know is outside the migration life history, outside of the breeding phase, when, let's make the comparisons when the birds are actually the most similar. Now, if you're doing a common garden experiment, it's not a problem because you're putting them in the same conditions. So what we were trying to do here is to find those stages that would be the most meaningful. And so we compared birds during the winter phase, post-breeding, which it was a little bit difficult with the Nuttles because their molt is much more slow over time, but much more extended. And then during fall. So those are the 3 stages we use to compare the migrants and residents. Now let's go through the same array of data. If we look at body mass, body condition throughout the common electricity stages, what pops out is that we're picking up an increase in mass in January among the residents over the other 2 stages. No change across the migrants. So it's more of a quiet time for the migrants, but for the residents, something is going on in January. If you'll recall, there's a time when this female showed very high baseline levels. If we look at fat store, there's not much going. You just don't see the amount of fat deposits that you do during migration or across the residents. So let's look at the pectoralis measures, and what pops out. This is what pops out, is that the fiber areas of the residents exceed that of the migrants. Migrants are unchanging and lower. The residents are not changing across the stages, but that's significant. If we look at the comparison of pectoralis in the migrant and pectoralis in the resident, they're different, that the residents are larger. I know it's a little bit hard to see that, so let's just look at the frequency distribution. If you look at the distribution of fibers, the residents are larger than that of the migrants. Let's think, well, if migrants are lighter, don't they need large fibers? Get back to that in a minute. If we look at pec mass, there's no real change across the migrants, but in the residents, we're seeing greater pec mass in January over the other 2 stages. So something definitely is going on in January with the residents. So thinking about the comparative traits across these fiber sizes, at first it seemed quite confusing. Because if you look at a larger fiber, what does that mean? We have more myofibrils, so each contraction is going to be stronger. They have lower resting metabolic rate, that is, the fiber itself is based on surface to volume ratio, which in a larger fiber is smaller. A smaller fiber, it's going to be larger. So having a larger fiber is actually reduced energetic cost across the membrane to maintain membrane itself. And what is coming to the fore now with in Gabriel Jimenez and his lab is this increase in myonuclear domain, which is where satellite cells move into the fibers and actually increase the ratio between the satellite cells and the cytoplasm, so you have greater potential for protein synthesis. So increase myonuclear domain. So it looks like migrants are kind of losing out. But when you think about it, smaller fibers relative across these 2 subspecies, you get increased vascularization. So smaller fiber has more vessels serving it. So that increases the amount of blood, the amount of oxygen, the amount of nutrients that are going to be received by the fibers themselves. So the interesting thing is that a number of studies that have done this, and Lundgren and Kessling did it, they were looking across a whole array of migrants. And they found that the migrants that were traveling greater distances actually had smaller fibers than the shorter distance migrants. And here, similar, we have comparison between the non-migrant and the migrant. And Jimenez and Messina are seeing a change in fiber size in preparation for migration, which we have seen as well. So let's look now at the power and fuel for migration and looking at the muscle profile. Residents have greater profile in January than migrants. Migrants are not changing over time. And muscle color, the biggest thing here is that the migrants are not changing, but the residents show a decrease or a pinker muscle around post-breeding. And talking with Jesse and with John and their studies on during the breeding phase for nodes and gambles found that fledglings leaving the nest leave with very big muscles. They're not particularly fat, but their muscles are very big. So this is an indication that this pinkness of muscle might not just be a migratory feature, but might be occur at a time when young or migrants are taking off for a period where feeding might be uncertain or unpredictable. Let's look at the Gastric Medius, migrant and resident, and what we have here is that across the residents there's no change, but we're seeing some slight changes among the migrants. And in terms of gastric medius mass, they increase that we have an greater mass in the residents than in the migrants. I think it'd be right if we look at the distribution, frequency distribution of migrants and residents in terms of gastric medius fiber area. Again, residents have larger fibers. They have larger legs. They have larger fiber areas. And the last feature, the last parameter is heart mass, and here migrants outstrip residents. They have larger hearts, and they're not changing over time, but the residents' hearts are small. Okay, so do the subspecies different? If we look at the life history states, residents have lower finite state. They have fewer stages per year than the migrants. So they are less constrained in terms of timing and duration of the stages across the animal cycle. Body mass, the residents are bigger birds. They have longer legs. They have larger pectoral fiber areas. They have larger fiber areas in the gastric medius. The fiber mass and the fiber area. For the migrants, they have longer pointier wings. It's one of the classic ways of identifying the migrant in the field, and they have greater heart mass. So let's just review the migrant resident comparisons. So the multiple life history stages, the HFSD constrains migrants in terms of timing and duration of the stages. Residents with fewer stages each have greater duration, the stages last for longer periods of time. Residents show greater variation across the stages compared to migrants. And those seem to appear during January. And we're thinking now that January might be the time when residents are most reactive or most responsive to local conditions. Residents are larger birds with generally larger traits, and the requirements of flight that have served to constrain migrants, affecting the size of their wings, their musculature, and size of the heart. So those sort of point areas that are going to be contributing to sustaining endurance flight have really been magnified across migrants over time. So in conclusion, is the expression of these traits the same or different? And what does this tell us about selective pressures for migrants? So the greatest differences we saw were in spring, as birds push north for breeding. Speed of travel, endocrine measures, power and fuel indices point to greater selective pressures on birds to depart early and arrive on the breeding grounds promptly, as the literature has suddenly documented that birds arriving early have great reproductive success. That doesn't mean that they have to get up there as fast as possible. outside the realm of what the conditions are. In some cases, if you arrive too early and conditions sour, as John showed yesterday, the chances of increasing reproductive success or even surviving. And the other caveat is arriving in a year like 2013, a snow apocalypse year, arriving early is not going to make any difference. You simply have to arrive, survive, and then initiate pregnancy. So it's not clearly and just get up as fast as you can. Get up as promptly as you can in the best shape you can. So, and are these traits the same or different across the migrants and the residents? And what does that tell us about migratory strategy? So the traits of the two subspecies differ during the life history stages they share. The duration and present stages are more prolonged, less time constrained than migrants, and more responsive to local conditions. That is, if conditions in January seem to be a period of time when residents are much more reactive in terms of their baseline, and females, particularly in terms of baseline levels, in terms of body mass, in terms of muscle mass. So for migrants, a long distance flight, so for migrants, long distance flight has affected both their timing and traits attributed to flight. And that's not the only thing. I mean, I think when people talk about this, it's not just the flight itself, but it is really the combination of that life history stage where you've got lots of stages, you're under time pressure to arrive, complete breathing and get out of the Arctic North, but also the constraints of just getting there, what you need, the parameters for flight itself, you create a larger heart, all the attendant features of maintaining a fast, efficient, long-lasting muscle. So, in conclusion, these morphological and physiological distinctions are indications or clues of the divisions, I think, that have pushed these subspecies into forging these migratory strategies. So you have one ancestral form over time, of course we don't know much about it, but then for whatever reason, the migrants kept moving and the residents stopped moving, and then all the selective pressures of those particular environments on these two genotypes or on these different species have pushed them further apart in terms of their physiology, behavior, morphology, and that all. So genetics probably not much. Okay. And I have to remember to do this because there are a lot of people that helped a lot, and I think our experience in Davis has been wonderful. A lot of postdocs, graduate students, and faculty have been just marvelous in terms of helping us get this project off the ground and moving into areas that I've never been involved in before. And then these undergrads, and wonderful, dedicated, and made it a lot of fun. And so, thank you very much for hanging in there. And that's it. Oh,
So with the muscle, when describe those two fast twitch muscle types, and one, I think this is gambled, I think, and one was really dominant in the winter, and then kind of became less.
Those are that myosin heavy chain.
Yeah,
the
those types. Yes. Okay. Sorry. The myosin heavy chain types. Do you think that might have something to do with like shivering thermogenesis versus the contraction that might be required for flight?
Exactly. And that's the same thing for the fiber size as well. Yeah, that's in fact, when people look at wintering birds, they're looking at fiber size as a function of the efficiency of the muscle to contract, which could be shivering thermogenesis or flight itself. So yeah, no, definitely. And when I talked to David's sponsor or reading his papers, his features associated with his northern parking well lines, those features sound like these birds are migrating. They fatten, they show hypertrophy. He's got measures on various muscle measures. So it's very similar.
And did you look in the Nuttles as well? Did they show the same kind of proportional changes?
No, we didn't.
Yeah.
It was a one-shot deal. So you guys, there you go.
More passing of the.
Right. Well, Randy has moved on, but Ken Walsh is very involved. He's at Scarborough. He was the one here in Scarborough. And he has a very, very active muscle lab. He did a PhD with Raul.
It's okay.
And he was in California at Santa Barbara.
Okay.
He was a whole Oxford student, so he came out of that whole chemistry school. But, no, I think that nice and heavy chains, and a lot of muscle people look at this a lot.
Yeah.
Right.
I had a question come through on the Zoom. And it's Sue, okay, says, do resident birds males defend territories all year, i.e. expend energy defending territories? Would that start to explain larger pectoralis, et cetera?
I think they definitely are territorial when they arrive in the, oh, wait a minute, this is Nuttles. This is, Nuttles. So it is interesting with the Nuttles that, after breeding, so one thing about Nuttles is that unlike the Gambles, the Nuttles retain their juvenile plumage, their womb, the juvenile crown for another year. And so on the breeding grounds, they, post-breeding, the juveniles with brown crowns form these large flocks and they move around. They're not territorial, but you have adults, but actually some juveniles that are territorial and they maintain those territories throughout the year. But then by the time breeding starts, everybody starts to carve out. So there is territoriality year round, but not by everybody. So what did she ask? Yeah.
I think she asked if that would start to explain larger pectoralis and stuff.
Well, I think that just the birds themselves, because the muscle data largely came from adults. I just thought we didn't have all that many samples, so it focused. But you know, when you look at juveniles, they weigh about the same. And so I don't think that that's an age thing. And it's really, I mean, they don't fly that much. So I think it has a lot to do with muscle maintenance that is just less costly to maintain larger fibers, which is kind of complicated.
I was also going to ask about sugar and thermogenesis,
Yeah.
since that's since pectoralis are generally the fuel for that
Yeah.
behavior. So Jamie preempted my question, but her version was much better formed than mine.
Okay.
But my backup question is, how many of these traits, if any, have you or anyone measured in Oriental-like crowns? Because it seems like a really interesting comparison because they have both elements of long distance migration, but they're presumably also altitudinal migrants, so they need probably even more flexibility and plasticity in terms of all these traits and behaviors.
Morton, of course, has done so much, but in terms of these measures, I don't think, I mean, he would have done the morphological differences, but in terms of fiber pecking and all of that, I don't think he has. But any system we have variation in terms of distance and length and altitude movement, it's going to be really interesting.
Yeah.
And the timing of it is so interesting. How this gets started, I mean, in this huge controversy in the field of when muscle hypertrophy begins. And I just think that people are measuring different types of, they're looking at mass, or looking at this. And so these things are changing differently. Now, why? The fibers are increasing, but we're not picking up a change in mass for some time, or the score for some time. So there is a lot of stuff in there. And talking with the muscle people, they were not at all surprised when we saw these increased muscle mass at arrival. I thought it's training, and they said, well, yeah, it is training, but you know, everything is with them. It's associated with the gym. When you go to the gym and you're exercising, your muscles increase, but a lot of that is fluid. So, and we didn't do dry muscle mass, which we probably should have done. I didn't answer.
Can we actually, like, totally on time, we're up against another class.
I saw people outside.
Yeah, I'm sorry, guys, we